สวัสดีค่ะ Good morning everyone. Welcome to the first International Joint Symposium on Agriculture and Food Security. My name is Pansiri Suthirak and this is Dr. Nataya Khonsu. And it's our pleasure to be a moderator for the beginning session of this symposium. Dr. Nataya will introduce the symposium and participants, please.
Okay, so this symposium is the annual symposium that organized by our school, the School of Agro-Industry. But this is the first time that we organize it virtually, so we call it the first International Joint Symposium under the topic of the agriculture and food security. The reason why we have to do it virtually because of the, as you know, the COVID-19 that is the problem in the world right now, right? So the objective of the symposium basically is to share the progress of some research work in our school. We would like to share what are the activities that we're doing right now, and we also would like to hear from the partner university, what are they doing in their lab work. And so therefore, whatever the topic that related to agro-industry, including agriculture, post-harvest technology, food processing, functional food, packaging technology, and as well as waste utilization are welcome to join. As you can see that the topic in the program is quite broad, so it might not totally related to your work, but it's a good opportunity that you can learn the new things from your colleague, and also you can seek the collaboration to work together in the future. So I hope all of you are going to sit tight and stay with us to support the presentation of our colleagues throughout the symposium today. Today we have totally 7 countries to join the symposium. This not include Thailand. So 7 countries from around the world, and there are 72 participants that kindly put an effort to join us today, and we're going to hear from them. There are 2 invited speakers and 22 speakers. I hope all of them can make it today, because right now we get a confirmation from about 10. So because of the very short notification, we put announcement like last month, and we organized the symposium a month later. So it is very kind of you to join us in a very short notice like this, right? So next, Dr. Pansri will introduce you to the whole program of the symposium.
Okay, thank you very much, Dr. Nataya. This symposium will be held today until 5 p.m. After this, Dr. Nataya will introduce the rules for presenters and audiences by the welcoming speech from our dean. And we will have the presentation from the first invited speakers and 2 presenters until 10 a.m. The next session will start from 10:15 by second invited speakers, followed by 5 presenters until 12 o'clock. And we will have a lunch break. And the afternoon session will start from 1 p.m. until 2:30 p.m. with the 6 presenters. And the last session will start from 2:45 to 5 p.m. with 9 presenters. And we will end this seminar with the announcement for the Best Presentation Award and the group photo session. And that's all for the agenda of the day.
Okay, I have the problem with the screen right now, but I'm gonna keep talking.
Okay.
So before we start, I'm not sure that the dean is here right now yet.
He's already here.
Oh, he is already here, but I'm not gonna allow him to speak yet. Sorry, because I'm an MC, I can control everything. So before we go further, I would like to make a few announcements so that we have the same rules and then we can run the symposium smoothly. First of all, I would like to ask all of you to rename the account that when you join the Zoom. Can you please use your first name? Yes, please rename the Zoom account and use your first name. And then please put the microphone on mute all the time. However, if possible, can you please turn on the camera too? but mute the microphone, all right? And because this is an online symposium, the active participation is very important. So we are welcome the comments or suggestions or the questions from all the participants. If you have any question at all, you can leave the questions or the comments in the chat box. And if you would like to ask in person, you can just wait for the Q&A session, right? And also, what is it? The staff will try to make sure that order of presentation is run smoothly. So we're gonna send the direct message to the presenter 10 minutes before presentation time. If we cannot contact you, if you have any problem at all, please let us know. And if that actually the problem that you cannot show up or you're not ready, we're gonna have to ask the next presenter to swap the order of the presentation, right? And then you can come back later when you are ready and just let us know, right? And I would like to tell you that all the participants that join this symposium will get a certificate. So to make sure that the information that appears on the certificate, which is your name, is correct, we're gonna send you in the chat box the file name that please go in and check out the information that if it is correct or not. If not, please correct it for us and so that we can put your name correctly on the certificate. All right, and also we would like to hear from you after this. We're gonna upload the file or the evaluation form, and then please give us the feedback. Right, that's it for announcement right now. We're gonna move on to the next schedule.
Okay,ค่ะ. And to begin this seminar, we are pleased to have Associate Professor Dr. Rawdkuen, the Dean of School of Agro-Industry, Mapalung University, to deliver the welcoming speech. Please.
Okay, indeed. Good morning, everyone. My voice is so clear?
Crystal clear.
Okay. So, good morning, everyone from 7 countries, including Thai, with totally 8 countries and more than 60 participate today. Thank you very much for all of you to be here today. As Dr. Nataya already mentioned, the objective of the conference, the symposium, as well as the how can I say, the very short announcement. Actually, this activity we have no plan before, just only for our students, our postgrad students to share the ideas and the progress of their research work. But finally, we think that according to the COVID-19 pandemic, we need to have any other activities to activate our MOU and MOA. So that's why we make decision to announce for asking all of the participate country or organization to join with us today. That's it, the main point. According in the past, actually, we bring or we brought our students to how can I say make the symposium in any other country. Such as like Japan. We brought our postgrad students to do the joint symposium in some university in Japan. That's it. And some year we do on-site symposium, like a professor from Korea, they bring their student to make the small symposium at our campus. That's it, the commonly. But according to the pandemic of COVID-19, so we cannot do any kind of activity like that. So come to the decision that we need to do like the joint symposium via the online platform. So that's why this activity will generate within the day today. And thank you for all of the co-joint researcher and postgrad student that you will give a talk today and share your research work or any progress of the thesis or dissertation. I hope that in the near future we can come and join us at MFU, at Mapalung University. We are planning to organize the international conference, actually like every 4 years So the next year, like 2022, will be our on-site international conference. We hope that at that time, all of you gonna be here at MFU. We are waiting to welcome all of you to join us at that activity. So for today, I think that's it, because like we have very tight schedules, and we have like as Dr. Nadia mentioned, more than 40 participation that will give the presentation. So we have a very tight schedule. So before ending my welcome speech, I would like to thank all delegates for your time and effort. And I wish this symposium will provide a successful and productive result to all of you. So thank you very much, and please take care during the pandemic of the COVID-19. Thank you very much.
Thank you very much, the Dean, Associate Professor Dr. Saroat Rawdkuen, for the very warm welcome speech. Okay, so before we move on to the invited speaker, I would like to tell you that we are recording the symposium, and the video will be uploaded on the school website. We're going to give you the school website later on so that you can download the video later. Okay, and every session, because all of you are going to come and go according to the different time zone, we're going to ask you all to take a group photo in the end of each session before the break of the session. All right, and next I'm going to show you a little bit about the instruction, how we're going to do the presentation. Let me share the screen. Okay, I hope you can see this. Anyway, okay. So you have 15 for the presenter, not the invited speaker, all right? For the invited speaker, you have 30 minutes. And for the presenter, you have totally 15 minutes for presentation. 12 minutes, you can present your work, and 3 minutes for the Q&A. We're going to give you the warning the first time. After 10 minutes, you're going to hear this voice loud and clear. This means that you have only 2 minutes left for the presentation. And after 2 minutes, you're going to hear the second warning. Okay, and that Q&A going to start. And after you use up 15 minutes, you're going to hear the third warning like this. Okay, so if you hear this thing, it means that your time is up and you have to stop presentation. And we have a committee to evaluate your performance, and the best presentation award will be given to the best 3 presenters at the end of the program. Right, we're going to have an announcement ceremony, which is very glamorous. Please wait until that session comes, and maybe our dean will be so kind that not only that certificate, that the best presentation award will give you will be waived for the registration fee for the international conference that we are going to organize next year. Yes. So if you win this competition, you can come to join us next year for free. All right.
Just only the registration fee.
Yeah,
it's for free.
Free registration, but you have to bring yourself to Maryland University. All right. So that's it for the announcement. We're going to move on to our invited speaker session, right? Today, it is our honor to have Assistant Professor Dr. María Luisa T. Mason from Central Luzon State University, the Philippines. Dr. María graduated PhD degree from University of Miyazaki, Japan, and her interest is about the genetic diversity and ecology of disotrophic bacteria. And that is why today she's going to give us a talk about potential of indigenous Rhizobia to increase the yield of soybean in the Philippines. Please, everybody, welcome Dr. María from the Philippines, please. Good morning, Dr. Maria. Dr. Saroat, raise your hand. Yes.
Hello, good morning.
Okay.
Good morning.
Yes,
How are you?
Can you hear me loud and clear? Okay, thank you. I'm doing great. So I think Philippines is just 1 hour ahead from Thailand.
Okay.
So I shall share my screen.
Yes,
please.
please.
Okay, please let me share my screen for a while.
It is
Yes,
our great surprise that Dr. Maria joined us in this symposium because we never had any collaboration before.
Yes. I was in Thailand last 2018 for 1 week symposium as well at the Assoc University.
I see.
Yes, so we spent there, mostly we have, I think, during the daytime and during the weekdays, we have lecture symposiums inside the university. And on the weekend, we spend 3 days touring around the, we went to so many places. One is the Asiatic.
Asiatic.
Yes. So we went there and I think it's a National Library.
I see.
So, yeah, it was,
But
it was very
yeah,
nice.
right.
Yes, and we also had dinner at a floating restaurant.
I see.
Very accommodating. And I also had many Thai friends when I was studying in Japan.
Yep.
Okay. All right. If you are ready,
Yes, sure. So I'll start my presentation.
please.
Please.
Okay. So I'm not sure about the question and answer. So shall I finish my presentation first, or anyone can just raise their hands?
I think it can be depends on your needs. I mean,
Oh, okay.
if you are comfortable to answer the questions during
Okay,
your
sure.
talk.
So if anyone wants to have a question or any violent reaction, you can just raise your hand or call for my attention during the presentation, and I'll be so much willing to answer your question. Okay, so as stated, okay. So as stated, the topic of my presentation for this morning is the potential of soybean rhizobia to increase the yield of soybean in the Philippines. Actually, this research is part of my dissertation. When I was in Japan, I conducted research about soybean and rhizobia for 5 years straight, mainly about genetic diversity. So, okay. So for the introduction, I'm not sure if how many of the participants for this morning is familiar with rhizobia. So at least please let me do a quick background about my topic. So rhizobia are diazotrophs that exist in symbiosis with legumes to form nodules. So when we say nodule formation, if we try to uproot a part of a leguminous crop, so those crops that have beans on the pods, like in Japan, they always eat lots of soybean, like edamame, and so many soybean products. So the rhizobia, or rather the legume crops, for example, this look at this as a part of the soybean plant, like the root hair. So once the root hair secretes flavonoids, so flavonoids are compounds that has node D protein. So these protein are recognized by the bacteria. And then when this bacteria, the rhizobia, so the generic term for this bacteria are rhizobia, when they recognize the presence of the node D protein, that will lead to the activation of the node genes. And then these node genes will lead to the node factors to initiate the formation of nodules. So once the nodules are formed, so the bacteroid, there would be a cell multiplication, and then the nodules will be formed. And inside this root nodule, there is this process that we call as the nitrogen fixation. So I think most of us in agriculture sector is very familiar with nitrogen fixation. Responsible for these are the presence of the NIF genes on the bacteria. So let's get over with the Rhizobia. First, why I chose soybean. Because soybean in the world is very agronomically important, not only because it serves as a human food, because we know that many countries, not only in Asia, but all over the world, eat meat, okay? Especially livestock like buffalo, carabao, or in the Philippines, even though the carabao has a very thick meat, they love eating carabao. So in other countries, maybe all meat stock. So most of the time they feed soybean meal. And soybean is not only good for human consumption, for animal consumption, but it is also very good for biodiesel stock. So when we are talking about the topic for this symposium, which is agriculture and food security, so I think soybean is very important as a crop that will feed the world in the future, because currently, most of the people who eat rice are mainly focused on carbohydrates, and many people who become very health conscious focus on the consumption of protein, and soybean is very high in protein. Okay, so in soybean and in Rhizobia as well, soybean and Rhizobia, they are very compatible with each other. They have a mutual relationship, and many studies before stated that the most dominant Rhizobia that are compatible with the soybean are Bradyrhizobium and Cynorhizobium. So these are classified as slow growers and fast growers. So as a brief introduction, so why do we say that soybean and Rhizobia are very compatible? Because we have this Rhizobia, okay? So the indigenous Rhizobia in the soil and the introduced Rhizobia in the soil. So the Rhizobia in the soil would now lead to the secretion of the product that will be used by the plants. These are the nitrogen. Because even though we have an abundant supply of nitrogen in the atmosphere, 78%, so those are not available. So before those nitrogen in the atmosphere could be used by plants, it has to be converted first. And the conversion is known as nitrogen fixation. So after the nitrogen is fixed, then it would be able to benefit the plant. So what is the benefit of the bacteria from the plant? The plant on the afterwards would produce photosynthetic products, and these synthetic products are generally carbohydrates. So the carbohydrates would serve as the food for the bacteria, so they could multiply under the soil. Then after that, Then after that, so when we try to do inoculation or the introduction of useful Rhizobia to the soil, generally there are competition because of so many environmental factors. Like even if we try to apply useful Rhizobia, the soil factors, the climate, other management practices would serve to compete with the useful Rhizobia that we introduced in the presence of the already indigenous Rhizobia. So this is the reason why we have to understand the ecology of the Rhizobia before we do any inoculation. So in the world, there are so many studies that have been conducted with regards to the Rhizobia. So we can see here on this map, this is the Philippines. Okay, so there are so many studies conducted all over the parts of the world, like in the temperate regions of the United States and the temperate regions of Japan. And their results are almost not similar. Okay, there are very minimal similarity because of the climate. So you can see here other Asian countries. We have the Vietnam here because our research group have conducted studies on these locations. That's why I highlighted these locations. Okay. And the part of my dissertation, I've collected samples all over the Philippines, and we have published some literatures. So I just like to not inform, I just like to note that on this article that we have published in, that we have published in 2019, so we were able to find out that the condition of the soil, if it's flooded or non-flooded, generally affects the community of the Rhizobia. So we can see the other soil factors such as the clay, the amount of carbon, or the amount of salinity. So these other factors affects the community of the Rhizobia in the soil. So what does this mean? So this means that all the other agricultural practices that we do to the soil, it affects the performance of the bacteria. So if we try to increase the population of the beneficial bacteria, it will also increase the production of soybeans. Okay, so what is now the current problem in the Philippines? Because my title is the potential of the Rhizobia to increase the soybean yield in the Philippines. So currently in the Philippines, our self-sufficiency is less than 10% because of the very low production area and the very low production harvest. So you can see in this graph that for more than 2 decades, our production is very low. So what are the research gap now in the Philippines? So in the Philippine condition, these are the identified research gaps. So we have to identify local varieties with different RJ genotypes, the presence of the indigenous Rhizobia, the factors that affects the diversity of the Rhizobia, such as the agricultural factors and the environmental factors, as well as the influence of these microorganisms to the restoration of the soil, because we know for a fact that if you have a healthy soil, then you can produce healthy crops. Okay, so I'm not sure how long I have been talking, but please kind of remind me if I go over the time, okay? And then, of course, the importance of this research topic on the economy of the Philippines is that if we would be able to find very beneficial species of Rhizobia that could increase the production of soybean per unit area, therefore, we can increase the self-sufficiency of the country by even more than 10%, I think. So we can increase the production as well. Okay. So this research, it focused on the isolation of soybean Rhizobia in selected location of the Philippines. So you can see in here the map of the Philippine Islands. So the Philippines is composed of more than 7,000 islands, but not all of it are inhabited, okay? So you can see 3 color variation. The first color, the orange one, is the main group of island. We call it the Luzon Island. Not sure if any one of you have been here to the Philippines, but if you have any plan to come and visit, so kind of let me know. I can tour you around. Okay, and then the second color, the yellow color, is the Visayas region, the second region of the Philippines. So this region is very famous for water sports. There's so many foreigners going in this part. And the third group of island, as you can see, has the largest land area among all the group of islands in the Philippines. This is the Mindanao. So I collected soil samples from representing the 3 locations in the Luzon, in the Visayas area, and in the Mindanao area, because I wanted to isolate soybean Rhizobia that represents the whole country. So actually, this is just a part of the dissertation, because this whole study, the whole dissertation, I collected more than 12 locations in the country. So not focused only on these 3 locations. And after isolation, so I opted to characterize the isolated Rhizobia based on their distribution and symbiotic potential. I'm really sorry if you can hear some noise on my background, because currently it's raining heavily in my area. So these are the information on the study site. So you can see in here that the location in the Philippines, we have 3 locations, and these are the location with the environmental conditions, the average temperature, the elevation, the rainfall, as well as the soil condition during the time of sampling. And after that, so I have provided the information also for the soil analysis. So on these 3 locations, all of the 3 locations, the existing land use during the time of the collection were basically soybean. And 1 location was used for rice during the wet season. And you can see that the acidity of the soil is very ideal for growing soybean, because soybean can grow by 5.0 up to 6.5 when it comes to acidity. And the salinity level is also very low, which means that soybean could really grow well on those locations. So we have a very moderate percentage of carbon, very low nitrogen. So this indicates that there really is a need to increase the nitrogen content or the nitrogen stock in the soil, because we have a very low fertility status here. And the texture, silty clay loam, and clay. And the numbers here signifies the number of isolates that I collected per location. So I have 70 isolates from one location, second location, and only 63. The reason I only gathered 63 isolates from this location is that some nodules that I collected, I was not able to grow any isolate from them, not because of the media, but I think because the soil of this location has very low population of the Rhizobia. So this is the general methodology for the whole study, for the whole research that I conducted. So after collection, I grew the soybean in laboratory condition using culture pots. So we used, or I used, a local soybean variety from the Philippines and grown it in growth chamber for 4 weeks. So we controlled the temperature of the daytime and the nighttime. Then after that, so isolation using growth media. So I use yeast extract mannitol agar, and then incubated for culturing, and then it undergo DNA extraction and PCR analysis. So on this particular report, I only opted to report the 16S-rRNA gene, since it is the golden standard for reporting the bacterial phylogenetics. Then after the phylogenetics, so there goes the symbiotic evaluation that I would show later on. So here is the result for the phylogenetic analysis with the targeting the 16S-rRNA gene. So you can see here lots of samples. I didn't bother to single out the names of the isolates, because these are just these are the total of the 203 isolates that I collected. So you can see in here that majority of the isolates that I collected has nodulating ability. So that means they can be used to increase the yield of soybean. So 67% out of the total population, and unfortunately, I obtained 33%, or about 67 out of 203 isolates were not able to form nodules. So this experiment was conducted 3 times for 3 cropping seasons. So in order for us to really verify that the non-nodulating isolates, which was collected, they were not able to grow any soybean on those locations. And then next is the graphical representation of the data that I gathered. So we can see in here that according to the sequence analysis result, the location on the Luzon Island is dominated by the presence of Rhizobia japonicum, or Rhizobia diazoefficiens. So let me just note that in the world, Rhizobia diazoefficiens is the most studied species of Rhizobia, because this species is the model species for Rhizobia studies, because Rhizobia diazoefficiens, it has the highest nitrogen fixation ability for increasing the yield of soybean. And since 2006, so in Japan, the first study that was conducted here, in 2006, they were able to identify some genes in this species that could complete the denitrification of nitrogen. So that means, because we all know that atmosphere contains 78% nitrogen gas, but that gas cannot be used by plants. So for other microorganisms, if they convert nitrogen gas into other forms of nitrogen, such as nitrates or nitrites, they produce nitrous oxide because of incomplete denitrification. And we know that nitrous oxide is a very potent greenhouse gas. Okay, so that really affects not only the food security, but as well as the environmental degradation. So interestingly, they were able to identify that Bradyrhizobium diazoefficiens, after its complete denitrification, it can convert the nitrates back into nitrogen gas. So that means if in the soil there are so many abundant species of this kind, so nitrous oxide emission to the atmosphere would be limited. So that is very interesting result. Okay, so we can see in here that these results are different from each location, but then the summary of this result is that this very interesting species is dominating the 3 locations that were sampled. So that means we have lots of potential candidates to be used as inoculant for future production of soybean. Okay, and then aside from the 16S RRME gene, so I opted to show this result, although this was not part of the abstract I sent. This is the ITS region, or this is the region between the 16S ribosomal gene and the 23S ribosomal gene, because 16S is a gold standard for identifying the genus of the bacteria. However, if we want to identify the species or to verify the species of the organism, so we have to analyze other chromosomal genes. That is why I show this result. So you can see in here, one location in the Philippines is really similar with USDA 110. BD, as shown before, is Bradyrhizobium diazoefficiens. So this is the model species. I think this model species, this was first isolated in the United States. That is why the name USDA, so from the United States Department of Agriculture. So it is now being used worldwide, even in the researchers of Argentina, of China, and other countries that research on Rhizobia. So they use these species for comparison with their indigenous Rhizobia. Okay, so let's move on to the application of the research results. So as I said before, we on the abstract, I have shown that we have performed symbiotic tests, both in the laboratory and in the field. So you can see here in the laboratory that we have some significant results. So these results shows that the nitrogen fixation of some indigenous Rhizobia in the Philippines is higher than the other indigenous species as well. So in this data, we can now select which species we can use for further research. So we can use for field application. So after the verification of this result in the laboratory, this is for the data on the nitrogen fixation. Next would show the data on the symbiotic efficiency. So this result means that how efficient is the symbiosis, because generally the nitrogen fixation does not necessarily mean that the efficiency is high, because nitrogen fixation just quantify the amount of nitrogen that is fixed. It does not quantify how efficient is the symbiosis between the plant and the bacteria. So again, we can see in here that we have some indigenous Rhizobia that can be used for symbiotic efficiency test on the field in comparison with the model species. Okay. Then, so after the laboratory test, so this is now the data that shows the symbiotic potential of the selected isolates on the field. So we have conducted field trials with 4 replications, and we have applied 5 ml and 10 ml of the Rhizobia on each seed. So as a slurry, so we applied it, we planted the soybean seed and then we applied it on the soil as a slurry. So that means we have cultured the Rhizobia to make it into a slurry. And then, so we can see in here, although I failed to put the letter suffixes here, but we can see based from the statistical analysis that we conducted, so this yellow is the non-inoculated treatment. So that means we have not applied any bacterial isolate in this location. So we can see SDW stands for shoot dry weight, okay? And then RDW stands for the root dry weight. So the shoot dry weight, the highest shoot dry weight was obtained from the location on the Luzon Island, which is significantly higher than the non-inoculated plants. However, the two locations have comparable results. So they don't differ significantly from each other. Meanwhile, for the root dry weight, it's just almost comparable with each other. So there's no significant difference when we are talking about the influence of the inoculation on the root of the soybean. And then I think this is the second to the last slide. So this shows the symbiotic potential, or I call it the symbiotic potential because with this data, we can now infer, okay? We can now infer which isolate we want to use so we can increase the pod per plant, the number of pods per plant, because the number of pods per plant would mean that we would have higher production of soybean, not only for human consumption if we opt to harvest as a green pods, or we can use it for soybean meal if we opt to harvest the seed. So we can see, even if the shoot dry weight of the isolate in Luzon is higher than the shoot dry weight of the two, but then their yield increase is just similar to each other, which is significantly higher than the non-inoculated plants. So this could mean, not 100% sure because it has to be verified, this could mean that all these three isolates could be further utilized as a potential enhancer of soybean yield in the future. Okay, so this is the summary of the results. So based from the results, we have found that the most dominant genus in the three locations is Rhizobium, which is 82%, and all the plant parameters were significantly influenced by the inoculation of the indigenous Rhizobia, and we were able to obtain some potential novel species that we should, that we should identify, we should study or focus more in the future. And this isolate has high possibility to increase the yield of soybean for food security. So that's all for my presentation.
Thank you very much.
Thank you so much. I think I used most of the time.
That's very perfect time. We still have like 5 minutes for the questions. Right, so may I have the questions from the floor if anybody have any questions at all?
Or you can leave the questions in the chat box.
Yes.
So Dr. Maria, from the information that we got, actually the Philippines is an island, right? Lots and lots of islands. So you have a few lands for planting, isn't it correct?
Very small area for planting.
Yeah. So it is very interesting that you try to increase the productivity because of to make use of the area, the land in the country.
Yes, even though the land area is very small, the population is continuously growing. So we have to feed the growing population of the Philippines.
Yeah, so I think we have the same problem in Thailand because we import about 90% of the soy in foreign countries. Yeah, but the planting area in Thailand is quite big compared to the land area.
Yes.
So I think if we can like duplicate your work.
Yes. I'll be so much willing to do a research collaboration to anyone who is interested.
So can I ask the question that quite because I'm not in this.
Sure. Yes, please.
Yeah, it's because the Rhizobia can increase the nitrogen in in the soil, right?
Yes, continuously, if continuously inoculated.
So can we use it with some other plants, not the soybean?
Actually, Rhizobia is not only present in legumes, it's also present in cereals such as rice, vegetables. Research on vegetables are very limited, but mostly the research on Rhizobia are on leguminous crops and rice.
So it means that we can apply this.
Yes, you can use it for rice.
Yep.
Just a different genus of Rhizobia. I think the genus for rice is Rhizobium.
I see. I'm not sure anybody in Thailand used to do that. All right. Any questions from post-harvest technology students? Maybe it is more related to your field. Okay. มีคำถามไหมคะ Any questions at all? All right. So if not, we would like to thank you so much, Dr. Maria, for your very interesting presentation.
Thank you so much as well.
Yes.
Thank you.
If anybody at all would like to confer in person, you can send a direct message to Dr. Maria.
So I'll now stop the sharing of the screen.
Yes.
Yes.
Thank you.
So please stay with us so that we can have a group photo after the before coffee break session. All right. Thank you so much, Dr. Maria.
Okay. Thank you.
Okay. So the times is very perfect. So we're gonna move to the first presenters of today. Miss Maria, the Los Angeles Ray. Are you there?
Yes, I am. Hello.
Hello. Okay, so Maria is from Inter, Argentina, and you have about 12 minutes for your presentations and 3 minutes for the questions and answer parts. So please.
You may
Okay,
start.
I will share
You can share your screen. Yes.
Let me see here.
Okay.
It's loading. There it is. Can you see the whole presentation?
Yes. Perfect.
Well, good morning to you. Here it's almost midnight. So first of all, thank you so much for letting me participate in this event. My name, as you have already said, is María de Los Ángeles. I'm currently working on my PhD thesis in the National Agricultural Technology Institute in Buenos Aires, in Argentina. I have a degree in food science and technology, and have been working in food microbiology for the last 8 years, both in industry and in academia. Today, I'm going to talk about a part of my thesis, that is the use of PMA-Qpcr to evaluate, this is a technique I use to evaluate effectiveness of high pressure processing in beef burgers to inactivate E. coli 0157. So, first of all, as an introduction, what exactly is E. coli 0157 and why is it important for us? This bacteria is a pathogen related to contaminated food consumption, most commonly meat products, but also vegetables and dairy products. Shiga toxin producing E. coli is commonly associated to hemorrhagic colitis and hemolytic uremic syndrome. And the most prevalent serotype is 0157H7. In Argentina, there is, this is a problem, this bacteria is a problem really, because we have one of the highest pediatric incidence of hemolytic uremic syndrome So this disease, as some of you should certainly know, has no cure. There are only treatments for symptoms, and since the 80s, several outbreaks have been linked contaminated beef consumption with hemolytic uremic syndrome. So in order to eliminate these bacteria from food products, we have a lot of alternatives. Recently, some novel non-thermal technologies have been developed. And to assess these relatively new treatments' effectiveness in pathogen reduction, traditional plate counts methods are used. They have a limitation, that is, we cannot quantify viable but non-culturable bacteria. Also, they are time-consuming and generate a lot of dangerous residues. An alternative then is to use molecular techniques. One of them, the one I used in my thesis, is PMA-Qpcr, that consists in two-coupled techniques that allow real-time polymerase chain reaction to quantify DNA only from viable cells. What is PMA? PMA is a photoreactive dye that penetrates compromised membranes and binds irreversibly to DNA. When coupled with real-time PCR, it allows to quantify only viable cells with their intact membrane. So we use membrane integrity as a viability criterion. Here, there is a diagram when we can see that in absence of PMA, in absence of this dye, polymerase amplifies all DNA in the sample, and when we add the dye, only DNA from intact cells is copied. In my thesis, I applied this PMA technique to different food processing technologies, but I'm going to show you my results with the one I like the most, that is high pressure processing. It consists in treating food at very high pressure in flexible sealed packaging for short periods of time. In the right, we can see how an equipment is it like, and in the left, we have like the equivalence of how high is this pressure. This technology has minimal residues, does not need heat to achieve good microbiological results. This also minimizes chemical reactions, so the product after treatment remains with a fresh appearance. We can use high pressure processing to treat ready-to-eat meat products and extend considerably the shelf life of them. When we want to assess pathogen lethality or how effective this treatment is, it depends on various processing parameters like how high the pressure level is and holding time, also temperature, and of course, which food matrix I'm working with. So with all this in mind, the objective of my essays was to use PMA-Qpcr methodology to assess high pressure processing effectiveness to inactivate E. coli 0157 in beef burgers. And how did I do that? Well, I selected for the PCR UIDA gene as for quantification of E. coli because it is a single copy gene. So one copy detected by real-time PCR was equivalent to one single bacterial cell. I inoculate beef burgers containing 4.75% fat with a pool of 5 non-pathogenic E. coli 0157 strains. As I show you below, there is a diagram of the experimental design. As you can see, I used 2 different culture media to plate count. Later, I will explain you why. And this was in parallel with the PCR technique. So, what were the results? First, in this figure, I have the plate counts results. For control samples without high pressure treatment, the one with 0.1 MPa, and for samples treated at 400 and 600 MPa. Then we define lethality as a difference in counts of tryptoin soy agar between treated and untreated samples. And we define injury as the difference between tryptone soy agar and the McConkey agar counts for the same treatment. As we can see, as at 600 MPa, a 4.6 log CFU per milliliter lethal effect, sorry, not per milliliter, per gram of burger, was achieved, while injury was not significant in neither of the treatments. Then, for real-time quantification, first, we needed a validated standard curve, which I obtained using DNA from bacterial culture and then diluting. Then I used the CQ values from the beef burger samples obtained from the real-time PCR equipment to interpolate bacterial concentrations from this standard curve. Once converted to log CFU per milliliter, we could compare bacterial concentrations obtained from plate count with PMA-Qpcr and with Qpcr alone without the dye. Here in the table, we have those values. As we can see, without PMA, Qpcr alone could not differentiate dead cells, oh, sorry, and amplifies all DNA present in the sample. Then I had a few conclusions about this work. Here I'm showing you a picture that is not clear at all. I'm sorry, I couldn't have better pictures of this work because the plastic bags always were messing with me. The first one was a burger without pressure treatment. The second was at 400 MPa, and the third one was at 600. As you can see, these beef burgers were raw, they were not cooked. So there is a little color difference, but once cooked, you can see no difference at all. So first, I concluded that high pressure treatment at 600 MPa was the most effective to inactivate E. coli 0157 in beef burger samples. Then the Qpcr alone was incapable to distinguish between viable and non-viable cells in samples with mixed populations, in samples that had been treated for some antimicrobial treatment. Then PMA-Qpcr apparently overcame this limitation, allowing to quantify using a previously validated standard curve only viable and viable but non-culturable cells. But in samples treated at 600 MPa, the differences between plate counts and PMA-Qpcr values can be explained by the limits of detection of both methodologies. So, with low bacterial counts near this limit of detection, conventional plate counts was more accurate than PMA-Qpcr, although viable but non-culturable cells cannot be quantified by this method. So this was all for now. Thank you so much for your attention. And well, if you have any questions, please let me know. I have been talking like so fast, sorry. This is because it's late in the night, so I'm very awake right now.
Thank you very much, Maria, for your interesting presentation. So we still have 3 minutes left for the questions and answer part. Is there any questions from the participants?
Thank you. I have a question, if you will give me time, about high pressure processing. Thank you for your presentation. It was very wonderful. I just know that you have focused only on the microbial part. What about the chemical part? Because you are using very high pressure, 600 megapascal, and it will have some effect on the chemical quality of the product. So what is your, like, are you planning for another study, to conduct another study to visualize the chemical parameters? For example, there will be some oxidation products during the high pressure processing. There will be the change in the proteins and so on and so forth.
Yes, personally, I didn't assess the oxidation part, because that was something that a colleague of mine did for her thesis. I assessed the color changes, also the pH and texture and water losses with the cooking processing. Well, I have those results. I am now working to get them published, and because I have like the main goal of my thesis is this molecular technique development and this validation. Sorry, I can't find the words for this. Like I try to show you this part specifically, but yes, I also studied all those other parameters. Yes, it's a very high pressure. So it's amazing microbiologically speaking. It's amazing how effective is this high pressure treatment to meat products, when meat have a lot of bacteria and very interesting flora. And after treatment, it's fantastic, really.
Yeah, appreciated your work. And one more thing about the DNA damage by this process. Have you ever estimated how much is the DNA damage in this E. coli? Whatever is your strand for the test organism. Like, is there any effect of the high pressure processing on the DNA damage of this organism for its inactivation or something like that?
Not apparently, not because not in the literature I worked with. The main effect the main effect of this pressure, high pressure, on bacteria is at a membrane level. So that's why I could use PMA, because PMA can enter only membranes that are compromised, that are broken. For example, if I had used, I don't know, irradiation that affects DNA, or UV light that affects DNA, I couldn't use PMA, neither PCR, because yes, there is a damage.
So it's more likely the bacterial cell lysis that exactly facilitates you. Yeah. Thank you very much for your answers.
Thank you for your questions.
Thank you very much for the questions and answers. So we also have the high pressure processing machine here. So if in future, if you have, we can be the collaborators on the high pressure processing one. Okay, due to the limitations of time.
Yeah, we're gonna have to move. Thank you so much, Maria.
Thank you.
Now you can go to bed. Please stay with us. All right, thank you. So we're gonna move on to the next presenter. All right, Analía, Cecilia, Colletti, are you here with us?
Yes. Hi, everyone.
Yes.
Can I share my screen?
Yes, please.
So Analía will present in the topic of applications of gamma irradiations on minimally processed fruits and vegetables. Are you ready?
I don't allow share my screen.
Yes.
So after the presentation of Analía, we're gonna have about 15 minutes break. And then we come back to continue with the second invited speaker.
I can't share my screen.
All right, we got your file. Did you send us the presentation file?
Yes.
So we're gonna share the screen.
Is it okay? We share the screen and then you can present the work.
Yes, please.
Okay. Okay.
Okay.
So Analía.
Hang on a minute. Please wait a minute. We try. We share the screen, but nobody can see it.
Hi, everyone. I have connectivity problems here.
Yeah, we can see you.
Okay. Okay.
Okay.
Can you?
Yeah. We already see your screen. Okay. Okay.
Perfect. Well, good morning, everyone. Sorry for these problems. My name is Analía Colletti. I am a food engineer and a PhD student in science and technology. In this opportunity, I am going to present the work entitled Application of Gamma Irradiation on Minimal Processing Fruits and Vegetables. My work is carried out at the National Institute of Agricultural Technology located in Buenos Aires, Argentina. This institution has several lines of research, including the development of new technology for the production of food and plant and animal origin, economic, social, and development, environmental development, and the transfer of knowledge to producer and consumer. In ancient times, agriculture and farming were the only source of food. Then the production of raw materials began to provide enough calories to cover people's diets. Over the last few centuries, this has been changing towards a more varied diet that provides better nutritional aspects, until today, where we see a market search for specific food that covers certain nutritional aptitudes. In this segment, we can see the fruits and vegetables. The regular inclusion of fruits and vegetables in the daily diet is associated with a low risk of chronic diseases, including cancer, diabetes, and other autoimmune disorders. Nowadays, there are changing in consuming habits, in purchasing decisions directed towards more sustainable production, but also reaching more distant markets with the corresponding quality certification. FAO promulgated this year as the International Year of Fruits and Vegetables, and its objectives are raise awareness and direct political attention to the nutritional and health benefits of fruits and vegetable consumption, promoting diversity, balanced and healthy diets and lifestyle through fruit and vegetable consumption, reducing losses and waste of fruits and vegetables in food system, and sharing best practices on promotion and consumption, improving sustainability, supply chains, and capacity strengthening. Regarding losses, we know that 1/3 of the total production is wasted or lost. In fruits and vegetables, the biggest loss is seen in the processing and packaging stage, followed by distribution and logistics. This leads to lower incomes for producers and higher prices for consumers. To prevent these losses and to increase intake of this product in a convenient way, consumer demand for minimally processed fruits and vegetables, ready to eat or ready to prepare, which prove able to preserve the freshness, nutritional, and healthy properties of the original fresh products. Despite the benefits of minimally processed fruits and vegetables, some essential operations can destroy the effects of this product, including microbial growth, enzymatic browning, tissue softening, loss of water and nutrients, and the development of flavors. Among the technological alternatives to preserve these products, non-thermal technology like high pressure processing, gamma irradiation, ultrasound, cool electrifiers, and cold plasma, high particulate raised the attention of the industry and academia in view of its proven effectiveness to maintain their quality, safety, and freshness. Irradiation is a physical process that involves the exposure of food to the energy emitted by a source of ionizing radiation for a certain period of time. Gamma rays are emitted by radioisotopes of cobalt-60 or cesium-137 and constitute electromagnetic waves of very short wavelength and high frequency. The product absorbs a controlled amount of energy per unit mass, defined as dose, and the unit is the gray in the international system. Irradiation causes ionizing reactions, damage to nucleic acid of microorganisms, but radiolysis of water, the main component of fruits, also occurs. Irradiation of any food at a loss of less than 10 kilograms presents no toxicological hazard or any special nutritional or microbiological problems. These studies carried out are so evident that no further testing is necessary. It is said by World Health Organization last century. So gamma irradiation is a non-thermal technology widely studied and approved by different agencies, such at the World Health Organization, International Atomic Energy Agency, and the Food and Drug Administration. In fruits and vegetables, gamma irradiation is used to control fungal and bacterial post-harvest diseases, to prevent foodborne diseases, to inactivate insect and parasitize, to delay the sprouting in bulb and tubers, to slow down physiological processes associated with ripening and senescence, and to preserve sensory, nutritional, and nutraceutic quality. All of them allow a considerable extension of the shelf life of products. One advantage is that the food can be packaged prior to processing. To discuss the process, simply put, the packaged food enters the ionizing chamber for a varied period of time, which determines the dose required to achieve the objective. A prior dose mapping is carried out to determine the minimum and maximum dose. The most commonly used is rhizobia, is cobalt-60, given that during its disintegration, it emits gamma rays of great energy power. Other applications are coloration of gems, sterilization of medical products, hygienization, insect disinfectation, polymer modification, and food for astronauts. There are more 200 gamma irradiation facilities in the world. China leads in both production and consumption. In our country, we have 2 facilities. In this other map, we can see all the irradiation facilities, including X-ray, gamma, and EV equipment. About my PhD research studies, low doses were applied to minimally processed species, and as I show here the main results. Stable color parameters, tissue softening, stabilization of firmness during the shell life, inhibition, enzyme activity polyphenol oxidase, significant increase in total phenol content, increase in antioxidant capacity in treated pitches, induction of small heat shock proteins, increase the perception of positive sensory descriptor like pitch flavor and juiciness. Fung growth was prevented on treated pitches. Irradiation treatment applied at low doses in combination with the use of low oxygen permeability films have positive and synergic effects. So irradiation represents a promising technology for improving the quality and increase the shelf life of fresh products. Finally, there is a growing interest in the food industry in application of emerging technology to maintain or increase the bioactive compound content or the antioxidant activity of fresh products. Numerous current authors report that when a low dose of irradiation is applied to fruit and vegetables, several metabolic pathways are activated, producing enzymes that catalyze the biosynthesis of secondary metabolites, many of them with health-promoting properties. For example, increased expression and activity of the phenylalanine monolyase, which is key in phenolic biosynthesis. So we are currently conducting research to better understand the physiological and molecular mechanisms against irradiation-induced stress responses, as well as optimal processing conditions that stimulate the further accumulation of specific groups of antioxidant compounds in fruits and vegetables. Thank you very much, and I am grateful for this invitation.
Thank you so much, Analía. You still have...
Sorry for the connectivity problems.
It's okay. That happens all the time. So any questions from the participants about the irradiation? How about the work in MFU as an oil? Do we have any irradiation?
So we did some UVC irradiations with the minimally processed fruits. I have one question. Can you hear me, Analía?
I think she's gone or not.
Analía, can you hear us?
Here.
Okay.
Sorry.
It's okay. Do you have problems about the appearance qualities after irradiated?
You can repeat the question?
Yes. Since we apply the UVC irradiation with our fruits, our flesh cut fruits, we have a bit problems about the appearance. So I just want to ask you, do you have any problems about the appearance of the sample after irradiated with the gamma?
Do you refer the intake of this product by consumers? Do not?
Just only the sensory test in the lab scale.
Well, there are negative opinions by this technology, but this is changing every time, and I think that in the future it will bear more positive opinion for this technology in all the products.
Okay, thank you very much for your answer.
All
Is
right.
there any questions from the participants?
Okay, maybe it's about time. Thank you so much, Analía, for your presentation.
Sorry, thank you.
So before we having a coffee break, and this is for the on-site participant only, we prepare the coffee break for everyone. However, before we have a break, can we take a group photo, please? May I ask everyone to turn on the camera for now?
Ready?
Do we have to take them off and hold the breath?
Okay.
Good morning, Gabby.
Good morning, everyone. Happy to see you again.
See you again. Okay, everybody smile.
1, 2, 3, the first shot. We have to move to another. Okay, 1, 2, 3.
You can see I'm smiling.
Yeah.
You can see I'm smiling.
Okay, 1, 2, 3. Okay.
Is it
I think I get all, maybe.
okay? Thank you very much. So one more time.
Again.
Okay.
Thank you so much for all the presenters for the first session for in this symposium. So next we're gonna have a coffee break. Now it is 10:05.
We will start the next session from 10:15.
We have only 10 minutes break.